everyone, and welcome back to ResearchCon 2023. My name is Nicole Kohr. I am a 30-year-old cystic fibrosis patient, a bilateral lung transplant recipient, and tonight I am your moderator. So welcome to this session, which is called What's Bugging You? The infection session. Woo! So before we get started and before I present you to our fabulous researchers and clinicians, I wanted to go over just a few cystic fibrosis related housekeeping items. So the first is that this session is being recorded. So it will be available after the event on the CF Foundation YouTube channel, and you will be notified by email when the session recordings are available. Please take advantage of this. As someone who has to practice infection protocol all the time or can't necessarily attend these events at a certain time, I always use the CF Foundation YouTube channel to watch the videos later. Take full advantage. Two, we will be taking audience questions at the end of this session. So we encourage you to submit your questions at any time in the Q&A box under the Stage tab, which is on the right side of your screen. We won't be able to answer the questions about individual diagnoses or have conversations about information that you learned today in terms of one-on-one individualized treatments. But please feel free to take this information that you learned today and forward it to your care team. See if that's something that you want to discuss with them. We will do our best to answer as many questions as possible, but if we run out of time, we will give you further options at that time. 3, we will be using Mentimeter. I'm very excited about this one. We're going to use it to answer some polling questions throughout the presentation. So these questions will help us get an idea of who everyone is in the audience and what you're excited to learn about or discuss today. So the poll questions can be found again on the right side of your screen under the Mentimeter tab. Big fan of this. It's my new favorite thing. So next to the Q&A and chat, you will see Mentimeter. 4, if you experience any technical problems during the session, please visit the help desk on the left side of your screen. So left is for help, right is for everything else. We also want to remind everyone of our community guidelines. Please be kind, open-hearted, respectful of everyone's opinion, experiences, and please do not give any medical advice. If you need any staff support for any reason or you want to report a violation, we encourage you to visit the help desk for that as well. Okay, so now moving on to more of my story and my journey with infection prevention. So this summary here has a picture of my beautiful mug a few hours after my bilateral lung transplant surgery in 2019. So again, this was about an hour after I woke up, and I remember this moment very vividly. I would say this was the peak of my life experience and the reason that I am passionate about infection protocol, infection prevention. So just to preface my story a little bit, I was diagnosed with cystic fibrosis back in 1997. I was 5 years old. At the time, there were only 2 medications available for cystic fibrosis patients. The first one was Pulmozyme, and the second one, which I believe was released in 1997, so it was very new, was tobramycin. I also have a rocky relationship with tobramycin, who has left me with several nodes on my vocal cords and deaf on my left side. However, you have to balance the risks with the rewards, and tobramycin did help me clear several sinus and lung infections in my day. So the next bullet point talks about my mutations. All CF patients have 2 mutations, and both of mine happen to be extremely rare. Because they're rare, I'm very much in the minority of the CF community. It's back in my day of 1997, I was told that because I'm not fully Irish, and this was considered an Irish people's disease, that I'm unlikely to have cystic fibrosis. But research has come so far, and we have learned so much about the inclusion and diversity aspects of this disease. And so people with CF look like me too. And we have mutations that range from common to rare. And so luckily clinical trials are becoming available for rare minorities like me as well. Throughout my youth and early adulthood, I was hospitalized anywhere from 3 to 10 times per year. Minimum, it's usually a tune-up, we call it a tune-up, for 2 weeks. That's a 14-day use of antibiotic treatment. Sometimes I would go home on home IVs. It would depend on the rate of my decline, the type of infection. I remember in December of 2017, I was struggling with malnutrition and I had a gastroparesis diagnosis. At that point, I was in the hospital for 3 months. And yes, you guessed it, it was through Thanksgiving, Christmas, my birthday, and New Year's, but we've all been right there, right? You got to do what you got to do. Right after that, the next bullet point shows that I battled Mycobacterium abscessus in 2013. I was a junior in college. I was very accustomed to being riddled with infection, but this one kicked my butt. This was a big one. This was the one that my care team advised that I drop out of school for a period of time and just focus on my health and do round-the-clock IV antibiotics in lieu of pursuing the rest of my education. It wasn't the first time that I had been told my life was at risk. When I was diagnosed at age 5, the doctors told my mother and I that I wouldn't live to be 10, and I've been proving them wrong ever since. So I have science and infection prevention and medications to thank for that, which is why when we reach the next bullet point, my bilateral lung transplant on June 14th, 2019 was less of an emergency and more of a treatment plan, right? It is by no means a cure. However, my lung function right before my transplant was 11%. I weighed all of 79 pounds. And present day, I weigh approximately 120 pounds, and my FEV1 is 135%. So yay, science. The last bullet point says Pasteur Act, AMR spokesperson, and volunteer engagement chair. We'll expand more on that later, but just to give a brief summary, the Pasteur Act is something that is being passed back into Congress today. Yay. It is a bipartisan bill that will help us with the drug development pipeline of antibiotics that are available to patients like me. And AMR stands for antimicrobial resistance. It was one of the many reasons I qualified for transplant, complete antibiotic resistance. So I'm passionate about that as well. And other ways that you can volunteer, we will share that throughout the program as well. Here I want to show you a little picture journal of my infection prevention timeline. So that first picture is what I already described of being diagnosed in 1997. I was in my very 80s, 90s kitchen with all the wood in the back doing a nebulizer. That was Pulmzyme and albuterol. The next picture is me with my mother, AKA my best friend, my care team, my support system, my everything. She endured every hospital stay with me, no matter how long I was there. And my walls were always wallpapered with get well cards from friends, family, and schoolmates. The next is one of my many sinus surgeries, the first of 4. After that, you'll see me on a train going to NYC, but I was simultaneously running a home IV. I had to do that a lot. I've been given a lot of feedback in terms of, is it safe to practice self-care with an IV on a train where it's riddled with germs and you never know what's in the air, but you have to live life in between, right? So I would glove, I would mask, I would take as much prevention and protocol as possible to stay well. The next picture is clinical trials. I've done many, we'll discuss that as well. The next is when I graduated from college, top of my class. When I had Mycobacterium abscessus, I decided that I didn't want to drop out of school. So I stayed in, I did 3 round-the-clock IV antibiotics until I graduated on time, top of my class. And there's no one size fits all when it comes to your journey with infection prevention. Everyone has different limits, different restrictions, different comfort levels. And so I wanted to finish my education, but in doing so, my lung function declined 35%. You know, doing 100% of everything isn't necessarily the way to go either. So you have to do what's right for you and listen to your body, right? After that, you'll see a picture of my nodes on my vocal cords. So that was one of my sinus surgeries, thanks to years of coughing and just irritation in my throat. The next is when I was working. I would spend my lunch hour resting or eating carbs because I was losing weight so quickly. My coworker took that picture. There were times at my desk where I would have a beaker, and I would use that beaker to measure the amount of hemoptysis or coughed blood. So it wasn't very healthy near the end. Luckily, I was able to go on short-term, then long-term, then Social Security disability, which is why I'm able to do events like this now. Following that, you'll see my feeding tube, a fungal infection, when my hair receded due to just malnutrition and medication, and that all of that caused hair loss. I've had hair loss many times. I'm very thankful for whatever's on my head. After that, you'll see my wedding, which took place on June 1st, 2019, a mere 13 days before my wedding. And that's my flower girl, AKA my oxygen tank, whose name was O2D2. And Disney, when you decide that I'm going to be the next Disney princess, please make him my sidekick. After that, you'll see me walking. That is my transplant surgery, and I walked within 72 hours. After that is when my husband, mother, 2 dogs, and I moved from New Jersey to North Carolina for a healthier lifestyle next to the beach. Lots of salt air. Natural remedies, right? And that last one is a picture of me being a foster mother and continuing to practice infection prevention simply because life goes on and we have to live while we survive, right? So while I give a few housekeeping items, I'll leave this slide here. You can see some of my post-transplant infection prevention techniques while I kick off our first presenter's presentation. So we would like to start with a poll for the audience. Please go to the Mentimeter tab now. And on the right side of your screen, it will tell us which of the following you've heard of. Have you heard of phage therapy, gallium therapy, and the CFF Infection Research Initiative? We'll wait a few moments now for everyone to respond. Let's give it like 15, 20 seconds, shall we? Boo, 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 boo. Boo, 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 boo. Boo, 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 boo. All right, it looks like we have a bunch of votes in. Oop, they're still coming. All right, let's give it another few seconds. That's so exciting. I'm seeing the bar going up and up and up and up. I'm so, okay. As someone who is passionately advocating for everything on social media all the time, it's very exciting when people have knowledge of these presentations coming in. So we have more to discuss. That's very exciting. Okay, so now we get to introduce our first presenter. Dr. Jennifer Baumberger is a professor of microbiology and immunology at the Geisel School of Medicine at Dartmouth, where she is also the associate director of the DART CF Research Development Program. She is a microbiologist that has been studying respiratory infections in CF for over 15 years, focused on how chronic bacterial infections get established and the impact of viral bacterial infections in CF. So tonight, she'll be telling us about her work on the Infection Research Initiative and exciting new antimicrobial therapies on the horizon in CF. What's up, Jen? How you doing?
Thank you so much for that invitation, Nicole. I'm so pleased to be here and honored to be here tonight for this session. So I'm really excited by looking at the poll data and seeing that there's already a number of folks that are well aware of the 2 therapies I'm going to talk about tonight, as well as the Infection Research Initiative. But I do see I still have some work to do too for others. And so I hope through this session to tell you about the Infection Research Initiative and about these 2 new therapies that I'm really excited about for CF.
Absolutely. Well, we're all looking forward to it. So take it away, Jen.
Thank you. So by way of disclosure, I need to disclose that I've co-chaired the Cystic Fibrosis Foundation's Infection Research Initiative, which I'm going to talk about here tonight. And my laboratory also receives funds from the CF Foundation to study bacteriophage and how they interact with cells in the respiratory tract in CF. So as Nicole's introduction so movingly illustrated, infections have been and remain an important concern for those living with cystic fibrosis. In the graph that you can see here on the right-hand side, you can see that over the years, patients with cystic fibrosis encounter a number of different bacterial infections, with all of us at this session are well aware of. The different colored lines here represent different bacteria, and the y-axis is the percentage of individuals. And so, as you can see, over the years, a number of bacterial infections are prevalent in individuals with cystic fibrosis. During the rollout of highly effective modulator therapy in the past few years, we've all been really excited to see the data that some of this bacterial burden appears to be, bacterial counts appear to be dropping. Some CF pathogen culturing appears to be dropping. So that's very hopeful and very exciting. But as this graph also shows, some of these infections are still persisting. And so it's with this knowledge that served as the impetus for the CF Foundation several years ago to initiate $100 million infection research initiative. And so this was to tackle infections in cystic fibrosis. And so the main goal and the mission of the CF Foundation's Infection Research Initiative was to spur new research and innovation in both the basic sciences as well as clinical research to drive forward new anti-infective therapies to treat infections in CF. And so to tackle this big issue, the CF Foundation brought together a steering committee that had broad expertise in infections. And so this committee was made up of a quarter of CF physicians, a quarter CF microbiology researchers, a quarter CF community members to give their perspective, and a quarter of the leadership from the CF Foundation. So together, we all worked together and came up with major goals for this initiative, which I'm showing down here in the bottom right left-hand corner. And I'm going to go over in a second. So the majority of our meetings were virtual because of the COVID-19 pandemic, but we were able to meet one time in person. And so the goals of this initiative were sixfold. And so I'm going to introduce each of those now. So the first was to continue investing in improving our understanding of CF microorganisms. And so this was funding basic science research that like my lab and others do in microbiology to try to understand how infections occur in CF and how they persist, with the goal of trying to prevent those in the future. In addition, trying to understand how infections are changing during the use of highly effective modulators like Trikafta, and what we need to know about those infections. The next goal was to improve our ability to detect and diagnose infections. As we all know, this is a major issue in CF, and it's only getting harder in this area of highly effective modulators because sputum levels are going down. And so sputum production has been so important for our ability to detect and diagnose infections. And so one of the goals of the initiative was really to drive innovation in this area to have new and more effective ways of detecting and diagnosing infections. We also put forth funding into areas that were aimed to optimize current treatments. And so taking already existing antimicrobial therapies and looking at new regimens or combinations that might improve the effectiveness of these current treatments in treating infections. And then a major goal of the Infection Research Initiative, and this was largely due to looking at community surveys over the years, and every time we're told again and again, develop new antimicrobial therapies. And so there was a whole lot of effort in this initiative into developing just that, new antimicrobial therapies. And I hope to show you some promising new leads on the next slide. And then finally, acknowledging this use of long-term use of antimicrobials in CF, and thinking about the side effects this cause and the development of multi-drug resistant organisms. And so doing research to try to better understand how we can limit those side effects and limit the development of antimicrobial resistance was another goal of the initiative. So as I mentioned, I'm gonna show you this the pipeline of antimicrobial therapies. And so this is the current landscape in terms of active anti-infective industry portfolio for the CF Foundation. And so each box is a different antimicrobial therapy and different stages in clinical development. I hope the very first thing you take away from this is really exciting. There's a ton of boxes here. And so there's a whole lot of work being done in a whole lot of different anti-infectives to try to improve our treatments for infection in CF. At the top in the blue bar, you can see this is showing you where they are in terms of clinical development. So discovery and preclinical, this kind of describes in vitro or animal-based studies in the laboratory to identify new therapeutics. Phase 1, 2, and 3, though, these are at the stage where these antimicrobial therapies have made it into patients and are now under trial in people. And so for both safety and in efficacy. And so it's exciting to see so many boxes in this category that are already at the stage of being tested in humans. The different colored boxes here represent the key pathogens that are actually being targeted by each of these therapies. And so you can look at your favorite organism or your organism you're battling and look at the therapies that are being developed. As you can see here, there's a lot of different therapeutics on this pipeline. I want to draw your attention and spend time today just talking about two main new therapies that I think are really exciting and want to talk to you about. And so those are bacteriophage therapy and the clinical trials that are being conducted, and gallium citrate. So let's start first with phage therapy. So first, what are bacteriophage? And so very simply put, bacteriophage are viruses for bacteria. And so these are very specialized viruses that kill specific strains of bacteria. Bacteriophages are found throughout the natural environment, and these are the natural predators for bacteria in the wild, if you want to think of it that way. It's important to note bacteriophages do not kill or replicate in human cells, and that's one of the beauties of these as a therapy, that they're really specific for bacterial targets. So then what is bacteriophage therapy? So phage therapy refers to the use of these bacteriophages I just described to treat infection in a person. Phage therapy has been delivered by both IV and nebulized in CF and other infection situations to treat in many cases, multi-drug resistant bacterial infections. And one of the really exciting features of bacteriophage therapy is that it's self-amplifying. And so what I mean by that is, if you look at this picture in the top right-hand side, we see that as a phage or bacteriophage interact with a particular bacterial target, so this is one of their targets that they're going to kill, they can inject their genome into the bacteria. The phage then goes on to replicate to many numbers of phage in that particular bacteria, eventually lysing and killing the bacteria, and that releases phage into the environment. In this case, we're talking about in the CF airway. And so the advantages there are twofold. So one, you've killed the bacteria, but two, you've made a number of bacteriophage, and each of those bacteriophage can now go on to infect a new bacteria. And then this process just keeps going on, and you can see how this is self-amplifying, that eventually it results in really impressive reductions in bacterial burden in the environment, in this case, the CF respiratory tract. So the one caveat that we need to acknowledge in phage therapy is that bacteria can evolve resistance to bacteriophage. And so one of the ways that many groups that study this have gotten around it is using a cocktail approach. And so this is taking several bacteriophage that target several different things in the bacteria and putting those together and treating a bacterial infection with all of them at the same time. And so basically that's making it much harder for the bacteria to try to evolve resistance to this one single, to numerous bacteriophage at the same time versus just one at a time. So if you take this cocktail approach to try to reduce the development of resistance, there's then two ways that are being trialed for this phage cocktail approach. One is using a fixed cocktail. And so this is choosing several different bacteriophage that are run through clinical development. And those bacteriophage, that combination can be given to a variety of different patients to treat infections with a particular microorganism, let's say Pseudomonas aeruginosa. The second way we could do this is a personalized phage cocktail approach. And so this is where your bacteria that is infecting your lungs would be screened against hundreds or thousands of bacteriophage and identify phage that can target your specific bacteria. And so this is, you can tell, much more time consuming because it requires time to screen these libraries and generate these phages. But those phage, that cocktail is really specific for your particular bacterial infection. And so these are two approaches that are being studied in clinical development now. So the CF Foundation has really thrown a lot of support behind phage therapy, which is really exciting. They've supported a number of different basic research labs like my own that study bacteriophage, how we might optimize these phage, and how the bacteriophage change microbial communities in the respiratory tract in CF. And then the really exciting part is they've also funded a number of clinical trials to study different companies' bacteriophage combinations. And you can see here that they're in various stages of clinical trial at the moment. I'm just showing on the right-hand side here several highlights of exciting bacteriophage kind of moments that have been in the news or in the literature in the past several years. And so this is one area I've been really excited about and I wanted to talk about today. So the second therapy I want to tell you about is gallium citrate. And so what is gallium? This is a metal. Gallium actually looks to bacteria a lot like iron, which I'm trying to depict over here on the right-hand side. When it's used in low concentrations in its salt form, it's safe, and it's actually being used currently in cancer therapy. And so it's exciting that it's already being used in people. So how is gallium antimicrobial? So gallium, as I said, it looks like iron to bacteria. And so when bacteria take this gallium up, they take it up through their iron uptake systems, just like they would iron. And the reason this is important is iron is absolutely essential to bacteria. It's important for growth and the function of many of their enzymes and metabolic enzymes. And so when bacteria incorporate iron into their enzymes, they work just fine. The bacteria can grow and replicate. But when they incorporate gallium into their enzymes, this prevents enzyme function and bacterial growth is limited. And so this is kind of the trick in gallium therapy, where we get the bacteria to take up gallium, they use it instead of iron, and it shuts their metabolic enzymes down and they're unable to grow. This is helped by the fact that CF sputum is actually iron limited. So when you either treat by IV or nebulize gallium, bacteria readily take it up because it looks like iron and they're in an iron limited environment. And so there's additional data that that's kind of coming around right now that gallium appears to be acting on immune cells to target inflammation. So we may actually be getting a bonus activity of gallium having anti-inflammatory effects, while we know it also has these antimicrobial effects. Two different features that make it an exciting new therapeutic. So I'm going to show you some data now from a study that was originally kind of the impetus to study gallium as an antimicrobial. And this is out of the group from University of Washington, Chris Gossett and Pradeep Singh. And so in this figure that I'm showing you, they took CF sputum and they basically treated this sputum with increasing levels of gallium and looked at bacterial growth over time. So testing this idea that if you give gallium, it prevents bacterial growth. And so if you look on the x-axis here, this is time over the course of a 20-hour period. And the y-axis is bacterial density, but this is basically, you can think of as bacterial growth. So the higher the level, the more bacterial growth. So you can see here in black with no gallium, the bacteria over time are growing. But if we give increasing levels of gallium, we see we can prevent bacterial growth, which is really exciting. The authors, though, acknowledge there is some iron in the respiratory tract of CF individuals. And so they did want to test also, can gallium be functional if there is some iron around for the bacteria? And so here they've treated this sputum with some iron. And you can see here in black, the bacteria like to grow in sputum that contains iron. But again, if they give increasing doses of gallium, they see they can completely prevent, and the purple and green lines can completely prevent bacterial growth. And so this was really exciting and led them to perform a phase 1 clinical study as part of this same publication. So this was a proof of concept study that actually at the end confirmed that the gallium was safe given IV. So the way this study worked was IV gallium was given for 5 days to patients, and then they were monitored for 28 days. And so when they looked at levels of gallium in the sputum, even though it was given IV, they saw sustained levels of sputum gallium for up to 28 days, which was fantastic. And then if you looked at their lung function, I'm showing you this graph here of over the course of the treatment, the 28 days, they see an increase in lung function as measured by FEV1 that peaks at 14 days and is sustained out through the 28 days of the study. And so this was really exciting data that led to what is now a phase 2 study that's underway, looking at nebulized gallium. And now they're actually looking to see if gallium has antimicrobial activities. And so those are just two of the many antimicrobials that are currently in development, but ones that I found very promising to add to our antimicrobial arsenal in CF and wanted to tell you about tonight. So with that, I'd like to turn the mic back over to Nicole. Thank you.
Thank you so much, Jen. My favorite part about your presentation is that it's like a Christmas movie. You know, it starts out with this hook because the first chart I see has 1991. Right. And so that's when most of my friends were born, in that 91 to 93 timeframe. So you hooked me right away. And then you say this scary emotional climax of emerging microorganisms, like there's new threats on the horizon. And now you have my attention more because now I'm all paranoid and fearful. But then you wrap it up with, but we have a drug development pipeline and our mechanism of action, and here's how we're going to do it. So like I feel like not only have you addressed these fears I didn't know I should have, but then you're already 10 steps ahead in terms of addressing them. So bravo. Thank you for everything that you do.
Yes, thank you so much.
Great. Okay, so now we are going to move on to our next presenter. Yay. And to kick things off, once again, please navigate to the Mentimeter tab on the right side of your screen and answer this poll question. Have you ever participated in a clinical trial of an investigational drug for people with CF? Let's see what our answers are. Do, 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 do. Do, 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 do. Maybe like 5 more seconds. I love this Mentimeter thing. This is very exciting. Okay, it seems like the polls stopped, so we're gonna cap it there. So now I get to introduce my new friend, Dr. Lisa Seaman is a professor of pediatrics at Columbia University Irving Medical Center and a hospital epidemiologist at New York Presbyterian Hospital. She has been participating in clinical research with people with CF focused on infections and infection control for over 25 years. As a member of the Cystic Fibrosis Foundation's Data Safety and Monitoring Board, she has a great appreciation and respect for the multiple ways that clinical trials are closely monitored for the safety of the participants. Today, she will be sharing her perspective on participating in clinical trials and safety monitoring before and during the clinical trials. How are you, Lisa?
Great, Nicole. Really thrilled to be here tonight.
That's great. What are your thoughts on these polls that you see?
Yeah, it's great. I mean, clearly, many people in the audience have been in a clinical trial, although many have not. And we have the opportunity tonight to really describe the way that safety is monitored, as you said. I just want to say at the onset that being in a clinical trial is not for everyone. And those who participate are very, very brave. And I'll give everybody a sneak peek that we're going to hear about your experience that you alluded to earlier. So with that,
absolutely, yeah. Take it away, Lisa.
Started. Oh, sorry, looks like we went to get to the beginning. Okay. So here are my disclosures. I do also receive funding from the CF Foundation for research and serve as a consultant for the CF Foundation. And as Nicole mentioned, I serve on the Cystic Fibrosis Foundation's Data Safety and Monitoring Board. Jen really outlined this beautifully, and I'll just reiterate that there are multiple phases that occur between a drug being thought about, never having been in people, and then it goes through a series of different phases that progressively involve more and more individuals until final approval, and then monitoring even after it's approved. So it's crucial that we think that the only way for our drugs to get into people with cystic fibrosis is actually to go through these clinical trials. However, the principal barriers to participating in clinical trials expressed by people with CF have been safety concerns. So we were really interested in better understanding how people with CF or the parents of children with CF understood safety monitoring. So we conducted a series of interviews with adults with CF, adolescents with CF, and parents. And what we learned was a variety of different reactions. Some individuals expressed that they had concerns about having a bad reaction. Since they didn't know if they were going to be given the placebo, meaning the sugar pill, or not, they decided not to do it. People are probably going to have problems during clinical trials. You don't want to be out on your own. But then others felt like, I feel like I am in the trusted hands of the people involved in the study. And it's not like there's only one person involved. There's a lot of people involved, so that they did feel supported. So we followed up that study with a second study where we actually recruited 150 individuals to look and see if their understanding of safety monitoring would actually make them more willing to volunteer to be in a clinical trial. And what we learned was that 95% agreed that safety was carefully monitored during a clinical trial. And 95% knew that they could stop being in the study if they wanted to. 65% were confident that it was safe to be in a CF clinical trial, and 65% felt that the benefits outweighed the risks. But what was actually surprising to us was that only about 20% of the people in this study understood the actual steps that were involved in the safety monitoring process. So I actually want to go through those with you tonight. So there are numerous safety steps for CF clinical trials, and I'm going to emphasize to start that these occur even before a single solitary person is enrolled in the study, and they continue throughout the study. The different layers of safety monitoring include the U.S. Food and Drug Administration, so all studies actually have to be filed with the FDA, and of course the study sponsor, who's responsible for designing the study and making sure that safety plan is put into place. Also, the Cystic Fibrosis Foundation has what's called the Therapeutics Development Network, or TDN, and they carefully conduct a review of all of the clinical trial protocols as well as the safety monitoring plan. The Data Safety Monitoring Board is assembled for each and every study, and I will, of course, talk more about that as well. Each study, a site, so every CF center that is conducting the study has to submit the study protocol and the safety monitoring plan to their individual institutional review board. Then, of course, the cystic fibrosis care team that is guiding the study at individual care sites. And finally, the person themselves that is involved in the study. So my own personal experience with CF clinical trials is that I had the great fortune of serving as the principal investigator for the azithromycin trials that were conducted in children, adolescents, and adults with cystic fibrosis. So I learned how to not only write the protocol, but to design the safety monitoring plan. As Nicole mentioned, I've been serving on the DSMB for 25 years. I'm one of the initial members of that. And as a CF clinical investigator, I've submitted numerous CF protocols to our institutional review board here at Columbia University. And I'm currently serving on the data safety monitoring committees of the two investigational drugs that Jen mentioned, gallium and phage therapy. So I just want to say a word about the data safety monitoring committees that are assembled for every cystic fibrosis clinical trial. DSMCs are an independent body. They are not the investigators. They are not members of the institutional review board. And on that data safety monitoring committee, there are physicians with expertise in cystic fibrosis. There are biostatisticians. There are people with CF, and there may be bioethicists and other individuals that are brought in as needed. For example, if there's a drug that there's concern about having liver issues, there may be a person with special expertise in liver issues. The DSMC knows which people in the study are getting the active drug and which people are getting the placebo. And they review every lab test, every pulmonary function test, every sign, every symptom that is occurring, and they conduct an independent assessment to decide if the study can go on as designed, or if there need to be any modifications made because they're concerned about safety. And in rare examples, there's so much concern that they may halt the study until things get addressed. So I'll just say a word more about the importance of individuals with CF who are actually participating in the study. It's crucial to take time to read the informed consent. It describes why the study is being done, what you'll be asked to do during the study, possible side effects of the study drug, and possible risks and benefits to volunteering to be in the study, reasons why you might choose to be in the study and reasons why you might not choose to be in the study. And again, I'll emphasize that participating in a clinical trial is really not for everyone. But during this pre-enrollment stage, this is an opportunity to ask any questions that you might have. And then during the study, it's crucial to follow the study plan, to take all of the doses of the drug, and of course, to let your CF care team know how you're feeling, including any side effects that you might be having, and communicate with your care team any concerns that you might have during the study. So I really was, as we've been getting to know each other in preparation for tonight's ResearchCon, we did learn that Nicole had actually experience with clinical trials. And I thought it would be a really wonderful opportunity to ask her to share with us her experience, which may not have always involved a study drug, I've learned, but she'll share that with you. And then also to tell us her perception of how her safety was monitored during the clinical studies. Thanks.
Absolutely. Yeah. Thank you so much, Lisa. Your presentation always makes me fangirl. I'm like, wow, like she's like, you've touched all the famous drugs, you know. But yeah, I've taken part in several clinical trials. The first was when shortly after I was diagnosed, actually, I was one of the first CF patients to do the vest. And no, not the Afflo vest where you can walk around and drive places, the one where there's like 1,000-pound machine and like a 1-foot tube and you hooked to the tube. So I did that for about a year, and my mother would monitor the endurance of the treatment, what level we put the vibration up to, what would expel more mucus, that compared to, say, manual chest PT. We did that for about a year. And, you know, although it definitely needed some testing, I strictly benefited from it. I mean, yes, I was certainly brave to be a part of that trial. Anyone does have to be brave to do so. But I felt like I was contributing to the betterment of our community. And in doing so, I was getting rewarded because I was getting treatment in the interim. So that was my first one. My second one was cannabis. So around the time when I was having gastroparesis and nutritional issues, cannabis was a way to possibly help with gaining or maintaining weight. And then the last one, I was lucky enough to take part in the phage study in 2018, shortly before my lung transplant. And I would say, just emotionally speaking, my participation in clinical trials ranged anywhere from it couldn't hurt to I'm desperate and I'm seeking this treatment as a last resort, right? And so I completely understand the emotional range that comes with a clinical trial. You're worried that something may make your symptoms worse, right? And I have to say, I had a wonderful experience in every single clinical trial. And of course, it's different for everyone, but I did feel like the communication lines were open both ways at all times. I had complete transparency, accessibility. They went out of their way to make me comfortable. They were flexible in terms of, hey, if you can't come on Tuesdays, let's change it to Thursdays. And let's give you a $25 gift card or stipend so that the transport isn't, weighing heavily on your wallet. And, I would just keep them updated with my symptoms. Like, hey, I started to have a headache, and then they would adjust the dosage. So in that way, I would argue there is nothing to be afraid of. It's only uphill from here. And, even if you are someone who receives a placebo, at least you contributed to the betterment of these studies. So I fully support it. I think it's fantastic.
Yeah. And that's such an important point you're making, Nicole, about that the placebo is necessary once we get into those later phases that Jen mentioned and I briefly described, because we have to make sure that if we're seeing symptoms, that we know that they are increased or not increased in the people that are getting the study drug versus the placebo. So, but it's also wonderful to hear about the experience that you had with transparency, because I feel like what you're really describing is a partnership between you and your CF care team. So thank you so much for sharing that.
Thank you for your whole presentation. I really appreciate it. Alrighty, so with that said, we are going to move on to our final poll and our final presenter. So we will all return to Mentimeter on the right side of your screen for the final time. And we'd like to know, what words come to mind when you think about this question? What kind of side effects have you experienced while taking antibiotics? So let's type in some symptoms that we've suffered through while on this lengthy list of medication. I'm very curious. We've got nausea, GI upset, loss of appetite. Absolutely. Those are good ones to start. As we keep entering in some symptoms, I'll give you a few seconds to think it through. I'm going to introduce our final presenter. So Dr. Joseph Rosano is a clinical pharmacist at the Yale New Haven Children's Hospital Cystic Fibrosis Care Center in New Haven, Connecticut. He has been practicing as a clinical pharmacist since 2017, and his interests in CF include infection, CFTR modulator therapy, and medication adherence and access. Tonight, he will be describing to us what roles the pharmacist, care team members, and those living with CF have in conducting good antimicrobial stewardship, and to get the best use out of all of the new and exciting anti-infective tools we have and available to us. So, Joe, what do you think of these words that you're seeing?
Thank you so much for that introduction, Nicole. And I have to say, you are absolutely killing it as our moderator. I think we got very lucky for this session with you. So thank you for getting us through the hour. When I look at our Mentimeter word cloud that's popping up here on our screen, what I really think and what's coming to mind first is that it really highlights that the patient experience with antibiotic therapies is really diverse and unique. And although we might define these and classify them as side effects, which they definitely are, in clinical practice as a practitioner, I tend to lean more towards characterizing some of these as true experiences on therapy, and everybody's is going to be unique. And that's going to ultimately play a part in determining a person's overall care plan for us when we decide on an antibiotic regimen that works for them. So my goal here today is really to showcase there's all these other variables as well that are going to come into place to drive our decision-making process when choosing an antibiotic regimen. And I'm really excited to share that with everybody today.
Absolutely. Well, take it away, Joe.
Thanks, Nicole. So as I mentioned, I'm here today to talk about all different types of variables that are going to drive the decision-making process when a care team chooses an antibiotic regimen. And that thoughtful decision-making process is called antibiotic stewardship. I have nothing to disclose for the remainder of my presentation, and I really want to kick off with working with you all to define what antibiotic stewardship is. The Center for Disease Control, or CDC for short, defines antibiotic stewardship as the effort to measure and improve how antibiotics are prescribed by clinicians and used by patients. Improving antibiotic prescribing and use is critical to effectively treat infections and protect patients from harms caused by unnecessary use and to ultimately combat antibiotic resistance. The CDC provides some key core elements to us that really embody what a successful antibiotic stewardship program will look like. Those elements include leadership commitment, accountability, pharmacy expertise, action, tracking and reporting, and education to the patient. Now, when we think about all the new and exciting anti-infective therapies that Jen presented earlier, we need to abide by one of my favorite quotes from my favorite superhero comic series, Spider-Man. And that is, with great power comes great responsibility. And that great power in our world of CF is going to be all of these new and exciting anti-infective tools that will be at our disposal. And that great responsibility is going to be how we ultimately choose to carefully and appropriately use these tools within the confines of good antibiotic stewardship. So to frame this in a way that's relative to us all, regardless of our knowledge of CF, regardless of our knowledge of antibiotics, and regardless of our knowledge of stewardship, let's talk about taking a trip to IKEA. Think of IKEA as being a drug company. And here we are as a community, ready and eager and willing to pick up all these new and exciting therapies that are in our drug pipeline, ready to hit the market. When we get home, filled with excitement about our new purchase, we sit down, open the box, pull out the directions, and then we're hit with that big pause moment. And in a fraction of a second, when we pause and stare at page 1 of the instruction manual, we realize we need all the right tools and parts to put this together correctly to get the best use out of it. And that, my friends, is exactly what antibiotic stewardship does for us. Antibiotic stewardship provides the tools to successfully equip practitioners and patients to garner the responsibility to use anti-infective therapies, i.e., our great power, most appropriately. The tools I want to review with you all today can be categorized into 5 buckets, and I like to call them the who, what, when, where, and the why and how of antibiotic stewardship. When we think about the who behind antibiotic stewardship, we think about the ever-evolving and dynamic CF care team. From physicians to nurses to researchers and other specialists, we all play a role and have a responsibility in ensuring proper use of antibiotic therapies. Now, I may be biased, but one care team member stands out to me in particular when I think about the glue that holds antibiotic stewardship together. And yes, that is your friendly neighborhood pharmacist, to keep the Spider-Man references going. I think if we think back to our core elements of antibiotic stewardship from the CDC, pharmacy expertise was a standalone recommendation as the drug expert. And effective antibiotic stewardship programs will have engagement from pharmacists on the teams, and some pharmacists can even co-lead antibiotic stewardships for an entire healthcare system. Pharmacists also have the opportunity to become board certified in infectious diseases, similar to how medical providers become board certified in specialty areas such as infectious disease and pulmonology. And for those of you listening, living with cystic fibrosis, if you are fortunate enough to have a pharmacist as part of your CF care team, then you may have experienced what it's like to have someone involved in the decision-making process behind which antibiotic regimen may be best suited for you. Now that we've defined who is involved in antibiotic stewardship, I want to discuss the what. One of the initial questions that will be asked when determining an antibiotic regimen is going to be, what exactly are we treating? And in order to answer this question, we need to marry 2 concepts together. What respiratory microorganism and where did it come from? Defining the respiratory microorganism by genus and species will begin to drive the conversation towards the appropriate antibiotic class that should be used. Is it a gram-positive organism, such as Staphylococcus aureus, or is it a gram-negative organism, such as Pseudomonas aeruginosa? You may have also heard some buzzwords from your day-to-day, such as strain, or second variety, or mucoid. These characteristics of the organism ultimately help to further define what type of infection we're really trying to treat. We also think about where the organism was cultured from, as we may have a different approach in deciding to start or even stop a treatment based on if the organism grew from a throat culture, a sputum culture, or a bronchoscopy. All in all, the CF Foundation does an excellent job in supporting care teams in this endeavor by tracking and tracing which organisms are growing across centers over time. And this type of data helps to frame the what of antibiotic stewardship. The tool crucial to the decision-making process within antibiotic stewardship is determining when to actually treat a person with CF with antibiotic therapy. This is critical to prevent overuse and unnecessary treatment durations of antibiotic therapies. Typically, the when will likely kick off with a sick call to the clinic. During the sick call, a person with CF or a caregiver will describe the symptoms that are presenting themselves, causing concern for infection. These symptoms can be changes in respiratory status, change in appetite or weight, and even describing how symptoms are impacting your ultimate activities of daily life for someone with CF. From there, the patient is typically instructed to get an updated respiratory culture, and the results will be reviewed in comparison to the previous culture results that we have from the past. Important questions will be asked, such as, is this the first time the person with CF has grown this organism, or have they grown it before chronically? Also understanding if they have recently been treated with antibiotics, and if so, which ones have we tried already? At what doses did we try it? And for how long have we already been treating for? All of which will help frame the decision of whether to start antibiotics immediately or possibly watch and wait alongside other interventions such as increasing airway clearance therapies or helping to address the underlying issues of any non-adherence to other medications that may be precipitating the symptoms that they're experiencing. Once we've determined it is appropriate to initiate antibiotic therapy, The care team will then be posed with the question of where to treat a patient to get the best results. When thinking about where to treat someone with antibiotic therapies, we have to think about that person's overall access to care, and social determinants of health may come into play. One question care teams may be commonly posed with is, can I receive these treatments at home in an outpatient setting, or do I have to be admitted? Variables such as cost for the site of care and insurance coverage can begin to come into play, and there may even be some instances in which therapy can be started inpatient and then transition to the home setting to complete the course. Another variable in determining where to treat is understanding and connecting with the patient on how their overall quality of life will be affected by this intervention. Are you going to miss school or work for a long period of time? Are you going to miss your support system if you're admitted? And if you are admitted, can your support system visit frequently enough so that there's minimal change? Lastly, it's important to be transparent that some antibiotic therapies require safe and effective monitoring by a pharmacist like myself, which we coin as therapeutic drug monitoring. This is the act of monitoring drug levels in the body via blood sampling to ensure doses are not too high, and on the flip side, not too low. Understanding if this type of monitoring is feasible in the care setting of choice is crucial to ensuring we are prescribing therapy safely to mitigate many of the side effects that you all highlighted at the beginning of my talk in the Mentimeter word cloud. Finally, we need to talk about the why and how. Arguably, the most important tool in our antibiotic stewardship toolbox. Defining our intent to treat and to use antibiotics is important to lay the foundation of why we are treating. Is the need for antibiotics due to an acute exacerbation, or is it necessary to continue to chronically suppress an organism to prevent disease flare-up? Filling in the details on how we treat a person with CF is one of my favorite parts of being a CF clinical pharmacist. Having the knowledge and connections with my patients and their families to know if they're going to actually be able to take and adhere to an oral therapy versus an intravenous therapy, for example, is of the utmost importance. As the saying goes, you can lead a horse to water, but you can't necessarily make it drink. In essence, I can prescribe a 4-year-old a liquid antibiotic, but if it isn't the right flavor, their guardian is going to call me within the next hour or so to ask for a new antibiotic care plan. Aside from the dosage formulation, I also have to take into consideration a term called bioavailability. To break that down, bioavailability of a drug is the amount of drug that will get to the site of action within the body that we need it to work. For example, when we think about it in our care setting and the site most important to us, the lungs, an oral form of an antibiotic may have a different bioavailability than the intravenous form or even the inhaled form. And thus, this will dictate how we ultimately administer antibiotics, whether it be by mouth, by intravenous route, or inhaled, or even a combination of the above. And to bring the conversation full circle, we must always consider our patient's experience with antibiotics. And answers to important questions, such as have you had previous side effects or intolerances, are necessary to ensure therapy is successful. Knowing a patient's previous cultures and their susceptibility patterns is also key to knowing if a bug may or may not actually respond to the therapy in mind. Lastly, knowing how a patient overall responded clinically to previous therapies will help determine if we use a similar regimen or try something new. And to give you a little glimpse of how we put antibiotic stewardship to work at my care center, we review our culture data annually and develop what is called an antibiogram. That helps us to frame not only the bugs our patients grow, but also the types of antibiotics we are prescribing over time. You'll see here, starting on the left-hand side of your screen, a number of different graphics that I've extrapolated for you all to give you an idea of what we track and trace as part of quality improvement in our care center. You'll see that we look at the number of cultures we collect annually, and of the cultures we collect, we look at the number of positive ones and assess which bugs we're growing. I practice primarily in a pediatric care center, so you'll see our major players here are staph and H. flu. We take a look at the culture source. So again, where is it coming from? What's the source? And then lastly, in the bottom of your screen, you'll see an excerpt of our antibiogram. And that antibiogram will show the percent of bugs that are susceptible or responsive to the antibiotic that we test them against. And that helps us to frame our decision-making and which antibiotics we choose when treating a patient. And while this data is very high level and representative of our entire care center, ultimately what matters most is taking all that information and applying it directly to a single patient. Individualizing antibiotic therapy is really the key to success. And we take all that information, combine it with the patient experience, and ultimately come up with a CF antibiotic action plan that works for the patient. And with that, I want to introduce Nicole back onto the screen, and we could take some time to hear about her experiences with antibiotics that she's been prescribed.
Yeah, honestly, Joe, you had me at Spider-Man, and then everything else was just chef's kiss, the cherry on top. So thank you for that. I found it so funny when you mentioned the flavor of the medicine and that they call within the hour. I did that well into my 20s. I mean, I would have itraconazole, and they would give me the liquid form because it was more potent, and I was like, I won't drink this. Yep. So like, you either give me the pill or I'm not taking anything. Like it
can be that simple sometimes. Yep.
Yeah. So it's so important. You see how like full circle this comes. So let's provide a real life scenario for you with my journey of antibiotics. Now, please don't take this chart too personally. Again, this was my journey with these infections and these medications. But if you're experiencing reactions similar to mine, just know that you are not alone. So for example, Bactrim has been mentioned. And so Bactrim is something that I take as a routine antibiotic. I'm on it to this day. In the past, it was used to treat MRSA and Stenotrophomonas maltophilia, a type of fungus that took me forever to not culture. My reaction to it was worsening gastroparesis and constipation, right? So that's something that is manageable in my journey with infection and antibiotics. But some other things were not quite as tolerable. Again, my journey with tobramycin, which helped to treat Pseudomonas, that left me deaf on my left side, vocal cord nodes, and eventually I became resistant. So now it's not even in my toolbox. If you browse the rest of my medication and reaction list here, you'll see resistance, hives, fevers, hair loss, migraines, diarrhea, a lot of the symptoms or side effects or characterizations, as you would say, Joe, that were presented at the top of your dialogue. And so I think one question I had for you, Joe, was I'm someone who looks at cff.org at least once a week because I like to keep up with all the articles and announcements. And so I often look at the pipeline as well. And often I sort it by what it treats, essentially. Do you find that there's a most popular or most sought out treatment style? Like, do you find people are more attracted to modulators in this day and age, or do you find it's still like, let's work on mucus clearance?
I think in the clinic setting, I think it's a decent blend. I think there's definitely interest in modulator therapy, but in the anti-infective realm, there are some patients who have very unique bugs and very hard to treat infections, and they want to know, for me, what's going to be personalized to me in the future. And I think that's something I really try to take the time with them to navigate on the CFF website and extract some of those upcoming studies of drugs that are coming in the pipeline that may be personable to them. They just don't know how to sort and search, as maybe not as savvy as you are, to find the right filters to find what they want to hear. But it's putting the information out for them and getting that to them and in their hands in the clinic and letting them know that these things are coming and they are in the pipeline, and it winds up being a very reassuring visit for everyone. So definitely excited for what's to come. Absolutely.
Joe, will you be my pharmacist? I would really appreciate that.
I will gladly be your pharmacist.
Well, how about you conclude our presentation, and then we will move on to some ways that our community can volunteer and do other engagement activities like we're doing now.
Yeah, so to round off our presentation with you all today, we want to highlight a couple key elements and then ways you can get involved. So to start wrapping up back from Jen's presentation, so the CFF Infection Research Initiative is supporting cutting-edge research and has expanded our pipeline to include a broad range of new therapies in development to tackle infections in CF. And we talked about bacteriophage and gallium.
Yep. And this one's called Community Voice. So to reiterate my little speech from the beginning, I am the volunteer engagement chair for the Central Eastern North Carolina chapter. I am also a member of Tomorrow's Leaders. I'm, again, a chair for that. I'm a congressional captain, as is Joe here. Both of us participated in the March on the Hill advocacy event in this past March, and really anything you can do, if it's liking and sharing a post from the Cystic Fibrosis Foundation on your social media, reaching out to your local chapter, there are many, many ways you can get involved, and nothing is too small or not enough. Whatever way you want to get involved is perfect. And one way you can do so is community voice. And so community voice is a way that it's a toolbox of resources, essentially, where you can find your talents and help assist the CF Foundation in whatever volunteer way you deem possible. I would say my first step, if I had to advise someone who's maybe new to the CF community, is to type in hashtag cystic fibrosis into Instagram or Facebook and just follow everyone who pops up, because that's the easiest way to be integrated into the CF family. And we will welcome you with open arms, right? Because storytelling is the most important part of patient advocacy. And so any way that you can tell your story, no matter what angle your story is coming from, we are all going to help find a cure for CF, right?
I love it. To round off our talk about clinical trials, and we really want to emphasize that to ensure the safety of people participating in clinical trials, there are numerous teams of experts who monitor them and seek to advance these new therapies in those with CF. And the safety monitoring occurs well before and during clinical trials.
That's right. And then, yeah, here's one example. I really like the clinical trial finder, because I'm always interested in something that can benefit me or benefit my friend who I know has a Delta F or things like that. So I'm excited to hear about this one.
Yeah, this is an awesome tool to use. So if you navigate over to cff.org/find, you'll be able to keyword search and filter and find all different types of clinical trials that are ongoing. And lastly, to round off my part of the conversation with you all today, in order to use all these new and exciting anti-infective therapies that are going to come to market, we need to appropriately and safely use them through means of antibiotic stewardship. And that's going to play a big role in CF care centers to promote the best outcomes for all those living with CF and to minimize any chance of antibiotic resistance.
Awesome. So again, these are a few ways we can take action, with a very exciting announcement also that has to be made. So let's say enter the PASTURE Act. This is a bipartisan bill that will essentially allow our drug development pipeline to be more accessible, more filled, more tools in our toolbox. So to read it verbatim, the PASTURE Act will establish a model to incentivize the development of new effective antibiotics and strengthen antibiotic stewardship programs. So if you're someone like me who is resistant to most or all antibiotics, that's the reason that I qualified for transplant. This is the bill for you. This will be life-changing for everyone across the board. And I'm very excited that it was reintroduced to Congress today.
Yes, big news.
Let's get it passed, everybody. Woo!
Very exciting and very timely for ResearchCon and our talk today.
Absolutely. Now, as far as our Q&A, we are going to start taking audience questions. So please remember to submit your questions in the Q&A box on the right side of your screen if you have not done so already, and make sure that you're in the stage tab. Okay, so the Q&A box that's available under the event tab is event-wide and not specific to this session. So we won't be able to see your question if you submit it there. Just make sure you're in the right box. We want to answer as many as we can. Okay, so let's see. Let me look at these questions. All right, first question. Which specific bacteria are being targeted by phage therapy? And are there currently any trials of phage therapy for NTM? Who wants to take that one?
Guess maybe I can jump in, and then if Lisa wants to add anything, since she is involved in some of these, the safety monitoring for some of these. And so a variety of different bacteria are being targeted by bacteriophage therapy. Each a specific phage being developed for a specific bacteria, as I said. And so a lot of them are being developed and run through trials for Pseudomonas aeruginosa and Staph aureus. Staphylococcus aureus. There are other smaller teams that are looking at other gram-negatives, but I think the main trials that are going on right now are in Pseudomonas aeruginosa. And then in terms of NTM, there are some compassionate use therapy or uses of phage therapy. Particularly the group at National Jewish in collaboration with folks at the University of Pittsburgh have been looking at have been treating some patients with phage therapy for NTM, but those are not in clinical trial at this point. And so, Lisa, I don't know if you have anything to add.
That's perfect.
Awesome. Well, thank you, Jen. All right, second question. This one's for Joe. How common is it for new adult patients to be put on antibiotics after their first culture? Because if the wait and see approach is chosen, how often would a culture be done?
Yeah, that's a great question. I think for us, it's really leaning into the patient-specific experience in that is there anything else going on with respect to adherence, maybe to airway clearance that led to an increase in symptoms, and other tools that we can provide them in that watch and wait period. If we do start antibiotics, it's usually that we're following one of our eradication protocols for some of those bugs that are first initial growers. So something like a Pseudomonas on first culture, we might approach it via an eradication protocol and still treat instead of watch and wait. But it really depends on each patient. When we think about reculturing, we usually try to reculture fairly soon after the symptoms present. So within the first couple days to a week of symptom onset, we're trying to get that sputum culture come in for that sick visit, reculture. And then, like I said, if we have any previous cultures, we're doing that cross comparison against culture history during the acute illness.
Awesome. Thank you for that, Joe. We have another one for Jen. So it says, does gallium also affect good bacteria?
Yeah, so that's a great question. So I don't know that I mean, so first we're going to define good bacteria. So I think I know what we mean. And so CF pathogens versus maybe commensals and other bacteria that are that we consider good bacteria are normal flora. And so one of the benefits of gallium is it does affect numerous different types of bacteria. Anybody who's taking up iron and using it will be affected by gallium. I think where this becomes beneficial maybe in CF, so it could affect good bacteria as well. What is known in the microbiology world, though, is that pathogens tend to be really good at taking up iron and competing for iron in low iron environments. And so in that way, gallium's a little sneaky in that it gets the bacteria, those pathogens that are the ones that are really good at taking up iron normally, they're going to take up more gallium. And so in a way, it can be more targeted for the pathogens in the respiratory tract. But I don't know that there's actual literature out there that's kind of putting together what is known and making my best guess.
Sure. Well, along the same lines, another question was saying, what does gallium do to those who are sensitive to metals, like jewelry and copper? Do we have any data on that?
I'm not aware of any data on that, Lisa, on the data safety monitoring. Did you guys?
No, and I can't remember that. I mean, that's probably like a different kind of sensitivity.
Yes.
That I would think like from skin, because some people do get a skin reaction. So yeah, it's a great question, but I bet it's different.
Okay. Well, then this one is for everyone. What advice would you give to a family with a 4-year-old who has multiple resistant bacteria growing in her lungs? And would it change in comparison to older ages?
I'm going to let the physicians take this.
Joe, you must have children like this that you're dealing with.
Trying to think what advice I give. I mean, I think the biggest thing for me is when we're talking to patients about the progression of disease when it comes to the ever evolving culture history, I think it's really important just to make sure you feel empowered to have those transparent conversations with your care team and understand what the differences in each culture really mean with respect to the susceptibility patterns that I was talking earlier. The data can be a lot to digest, but if you do vocalize and express that you want to understand it better and understand what does the change in that pattern mean for my child, those are all definitely fair questions to ask and to feel supported and encouraged to break down the science with your care team and understand what trajectory each bug is growing in and what that means for available therapies to be able to provide. It's very much a collaborative conversation that we have with patients when it comes to those types of situations.
Yeah, I love that. And I think you made that very clear in your IKEA reference, which I love. I'm going to carry that with me forever. All right, we've got one last question and then we are wrapping up. So what challenges do the rising numbers of pregnancies in people with CF pose in terms of treatment options? And this goes for a question in the comments as well of like, is anything targeting fungal specifically? So pregnancy and fungal, anything to say on those?
I think from just I can speak from a drug management standpoint. A lot of what we consider when using drugs in pregnancy are fall back on one of the terms I used earlier, which is bioavailability, right? We think about how will the drug potentially reach the unborn fetus? Will it actually cross the placenta? Is there a concentration sufficient enough to cause worry or potential harm? And we have those, again, honest conversations with each patient. We provide the transparency and the numbers around what % chance do we have of this drug potentially affecting the unborn child? And what does that look like in terms of alternative therapies if that risk is too high? There's also data for drugs out there to support what % of them are expressed in breast milk. So thinking about breastfeeding too, even post-pregnancy as well. So drugs and maternal conversations are very much a big part of the antibiotic conversation for sure, because there's all those variables that come into the conversation.
And it's fair to say that a lot is known, right? Because we know a lot about what is safe to take in pregnancy based on a huge body of literature that's been evolving over many, many years, independent of people with CF. So I think that can be very, very helpful in guiding people to what's safe during pregnancy.
That's awesome and so hopeful to hear. I feel like this whole presentation is like, here's where we once were, and now we're talking about pregnancy, right? That's fantastic. So as a patient with CF, thank you all so much for your contributions in research and clinical work and trials and everyday management. You are truly elongating lives like mine. So thank you. I appreciate it. All right, so we are just about out of time. I want to wrap up. And thank you to all of these speakers again for sharing their knowledge and insights about infections. Thank you all for your questions, your participation, your just watching us chat for over an hour, right? Before we go, please take some time to fill out the satisfaction question under the poll tab on the right-hand side of your screen, right next to the session checkout or the session chat. We'd love to know how you felt about today's conversation. Did you like it? Were there parts that stood out to you? There were also many wonderful resources related to this discussion. So including the slides from today in the resource document found on the event reception page, those will all be available to you. Please spend some time exploring the resources, downloading anything you want to keep. During today's session, we mentioned opportunities that are available through Community Voice. So if you are interested in learning more or signing up for Community Voice, we will drop a sign up link in the chat. So keep an eye out for that and click. It's very fun. I highly recommend it. Next up on the agenda is a 30-minute break before the closing keynote begins at 8:15 p.m. Eastern Time. Or if you would prefer to continue learning and don't need a break, we are offering virtual roundtables in the Sessions tab on the left-hand side of your screen. And these roundtables are staffed with a representative who can take your questions and provide you with more information on foundation-sponsored research projects, initiatives, etc., including the wellness resources available to the community. So attendees are encouraged to jump on, please. I'm going to be on one of the sessions as well. I'm happy to chat with you more. Share your mic, share your video. Let's interact directly. And thank you again for coming to our session. We are so thankful that we had an opportunity to give you all of this information and more here at ResearchCon this year. I hope you have quickly learned what's bugging you as of 2023. And thank you again for your time. Happy, healthy life, everyone. Happy, healthy night. And thank you for coming.